discussed a lot about the apparatus qualification and validation and all. So now I want to just spin it a little bit in a different direction. So like this morning in the introductory lecture by Dr. Vinod Shah, I think he kind of envisions or he tried to advocate a couple of things which caught my attention, and I want to ask the audience as well as the other panel members what they think about what he envisions in future direction. One of them, he kind of in proposing IVRT mandatory similar to oral dosage form dissolution. Number two, he wants to see IVRT as a quality control tool. And third, he also talks a little bit about TCS, and especially what caught my attention that he wants to use the IVRT for the approval of lower strength or different strength. So it is a little bit, we are deviating from science a little bit more towards the regulatory. So, but these comments are, I think, very much pertinent, and I want to hear that what audience think about it. Should we make the IVRT mandatory? Should we make it as a mandatory quality control tool? And should we use it for approval of different strengths? Thank you.
Well, let me offer my comment. I'm actually glad, Tapash, that you brought that up, because it's valuable for the same audience to have a perspective of, what are some of the considerations around those that vision. When it comes to the idea of using IVRT as a release specification or a stability specification, I think some of the concerns there are, especially on stability, that dosage forms do change. So across their shelf life, the viscosity may change, particle size may change, and an IVRT may be very sensitive to those changes, to the point that even though you've got a shelf life, and this is an established product with an established shelf life, now you're going to have product that's going to start to fail the IVRT. And I think one of the questions, and this had come up years ago, and the questions from a lot of our industry colleagues were, well, but there is no expectation of in vitro, in vivo correlation here. Might we just be failing perfectly good product? What does this mean? And I think the lack of evidence to be able to support that kind of change was very concerning to our industry colleagues. And it was a fair point here. And I think we don't have enough information to understand what the implications of that are to use it in that context. I think this is one of the concerns. One of the other concerns related to using it in bio waivers for different strengths of topical products. And the challenge here is, while I think in general IVRT can provide some information in the context that it's used for bioequivalence, where it's a constrained context where you know a lot, you don't even qualify for using it unless you know a great deal about the components, composition, arrangement of matter, and other features of that dosage form, and then you're mitigating certain failure modes for bioequivalence. I think there's some utility there that I think we can continue to parse out. But with the bio waivers for different strengths, topical products that occur in different strengths are not necessarily the same formulation at those different strengths. Sometimes it's just a completely different product. And so I think one has to be cautious about how one thinks about that. And for me, one of the fundamental, I don't want to call it a fatal flaw, but a fundamental issue with the TCS that is being proposed is that it proposes essentially to utilize IVRT as a surrogate for Q3 characterization. In other words, as long as the IVRT release rate is the same, then we assume that all of the arrangement of matter must be the same. But we have evidence, we know that's not the case. We at FDA have even published on this work where you can change the Q3 properties and still have a passing IVRT. So this is one of the problems, that the construct of the TCS, while very appealing, that unfortunately has some scientific problems in it, and this is not something that's being supported certainly by by anyone that I know within Well, I'll speak for myself that I would be cautious about interpreting that as FDA's position. I think that is the position of Dr. Shah, and hopefully we can have that distinguished. But it would be interesting, as you say, Tapash, to hear other people's opinions on things.
I have a concern about the concept of using IVRT for release and stability as well. And we have, when I was at Turgus, we had many times were approached by sponsors to develop IVRT method that can be used for release and stability. And my answer always has been that we really need a very large body of data to generate, a good database of IVRT testing of different lots on different times, different parameters that we don't even know about, so that we can build good specifications and criteria for pass-fail if it were to be used as a pass release. And for stability, I don't even know how one can really put any specification depending on the properties of the dosage form. So that is a great concern. What I would say about TCS is that I think the way I would phrase is that in some cases, IVRT might be able to look at the, while it's not true that anytime you have similar IVRT that you have Q3 similarity, but when you have Q3 similarity, then you will have IVRT similarity. The IVRT, your release rates are going to be, should be similar.